We discuss about lag culture, lag insects, Carrier laca. Lag producing scale insects that live on host plants. Proposes into plants and feeds on plant sap. Lag glands secrete resinous substance lag encases and products them. Reside inside retinous covering lag cells. Adult lag insects are small orange-red organisms. Adult female is wingless, inactive, and larger. Male is winged and actively moving. Female legs dice hatch out nymph carver swarming nymphal body differentiation nymph grow lac cells formations lac secretions molting of nymph 3 molts prepupal pupal stage adult lac cultivations. First one is selections and preparations of host trees. Second one is introductions of lac insects to host trees and harvesting of the lac. First one is host plants: mango, fig, peepal, gular, teak, oak, beer, acacia, etc. Quality depends on host plants. Pruning results in formations of numerous tender branches. 4 or 5 months after pruning, lac insects are introduced. Inoculations introductions of young one of lac insects to host trees. Natural inoculations occur normal part of life cycles of lac. Artificial inclusions lack encrusted wings with eggs and young ones are taken from a donor tree, broadlack and tied to pruned host tree. Longitudinal inoculations facilities makes contact between broad and host. Drawbacks of natural inoculations prepared infestations of the same tree by lac insects reduce vigor and vitality of tree and retard its growth need not be regular sequence warming larvae may settle on branches in a randomly dispersed manner greater change for multiplication of predators and parasites cope system of inoculations to avoid repeated infestations of the same tree. Host trees are divided into blocks based on variety, growth, and size. Each crop is inoculated according to definite time scheduled. Ensure max rest to host trees. Precautions for artificial inoculations. Wings and broadlap used for inoculation must contain good number of eggs but no predators and parasites. Nymph used for inoculation must be healthy and ready to war. Host trays should be changed for every inoculation. 4 or 5 wings must be used for every inoculation. Standing of lac insects, crops, kusumi, jelly 6 months. Cultivation period January to February. Harvested period June to July. Agani 6 months June to July. January to February. Harvesting period January to February. Rangini Kartika 4 months. Cultivation period June to July. Harvested period October to November. Vaishagi 4 months. October to November. Cultivation period October to November. Harvesting period June to July. Third one is lag harvesting. Collections of lag form from host trees for commercial use. Immature harvesting. Harvested before nymphal swarming. Harvested lag is called airy lag. Mature harvesting. After swarming lag is called mature lag. Immature lag harvesting is not preferred. Lag that remain attached to bark on of host trees, broad lag. It is cleared, crushed, and powdered, and then sun dried. Purified lag, grain slag. Grain lag is melted on charcoal fire with arsenic sulfate and then filtered. Immature lag, resid, kiri lag. Clear residue is cooled and solidified. Pure lag, button lag. Button lag is then made into thin sheets. Sheet lag. It is chemically proposed to form pressed from shellac used for commercial purpose. Composition of lag: resin, water, sugar, proteins, soluble salts, etc. Coloring pigments: lactics, dye, and erythrolacins. Properties of lag: water insoluble, soluble in alcohol and ammonia, bad conductors of heat, high active power, forms bending material when mixed with alcohol. Use of lag: shellac is used in paints, polish, varnishes, imitations, flowers, etc. Used as thermoplastics material. Manufacturers of glass, paper, optical frames, crayons, etc.